Hi, my name is Savannah Stelzer, and welcome to my presentation on hormonal control of implantation and embryonic development. Let's begin by discussing the process of implantation. Once an egg is fertilized by a sperm, it becomes what is called a zygote. This zygote begins to travel through the fallopian tube until it eventually reaches the uterus, which is its destination for implantation. While on this journey, the zygote begins the process of mitosis, or cell division. This occurs typically on days 2 through 4 after fertilization. In total, the journey from the fallopian tube to the uterus takes about 4 to 5 days. This is where we encounter the first hormone we will discuss in the process of implantation, progesterone. Progesterone plays a role in relaxing the smooth muscle walls of the fallopian tube, resulting in the slow travel of the zygote. When the zygote reaches the uterus, it becomes about a 100-cell unit known as a blastocyst. The blastocyst consists of an outer layer, which will eventually become the chorion and form the placenta, and an inner layer that will become the embryo and several additional and critical membranes. These membranes include the amnion, which secretes amniotic fluid the embryo floats in, the allantois, which becomes part of the umbilical cord, and the yolk sac. This blastocyst implants on the endometrium of the uterine wall sometime during days 5 through 9 after fertilization. This occurs by blastocyst secretion of an enzyme that results in the endometrium engulfing the blastocyst. Here is an image taken from Silverthorne that details the process of implantation. Here we have an egg that's released via ovulation, and it's fertilized by a sperm. Once fertilized, the egg becomes a zygote and begins to travel through the fallopian tube. All the while, it's undergoing mitosis during days 2 through 4 after fertilization. This zygote eventually reaches the uterus as a blastocyst, where it then implants on the uterine wall and begins to develop. That usually occurs on days 5 to 9 after fertilization. Once implanted, the embryo begins to develop. Throughout this process, we see several hormones, processes, and structures that work together to ensure proper implantation and development. One of these critical structures is the corpus luteum. The corpus luteum is a ruptured follicle that has been transformed into a structure that secretes hormones that are necessary in the early stages of pregnancy. One of the hormones secreted by the corpus luteum is progesterone, which plays many critical roles in menstruation and also in embryonic development. The corpus luteum can only survive for about 12 days unless an egg is fertilized. If an egg is not fertilized, the corpus luteum will disintegrate. This disintegration causes a drop in both estrogen and progesterone levels. This in turn results in menstruation and the excretion of the embryo from the body. If the embryo exits the body during menstruation, the pregnancy cannot continue. However, if an egg is fertilized and the blastocyst successfully implants on the uterine wall, the developing embryo can send a signal to the corpus luteum that will prevent its disintegration. This allows it to remain present for the first several weeks of pregnancy. The corpus luteum releases progesterone, which stimulates endometrial blood vessel growth and also nutrient secretion from various glands. The corpus luteum is under control of a hormone called human chorionic gonadotropin hormone, or HCG. Human chorionic gonadotropin hormone, or HCG, is secreted by the placenta and the chorionic villi. The chorionic villi are finger-like projections that sprout from the chorion and allow maximal contact with maternal blood. HCG is a peptide hormone, meaning it is composed of linked amino acids. This hormone is a close relative to luteinizing hormone, or LH. Because of its similarity to luteinizing hormone, HCG binds to LH receptors. The presence of this hormone allows the corpus luteum to continue releasing progesterone. Progesterone plays many critical roles, including keeping the endometrium of the uterine wall intact so the embryo may remain implanted and in turn can continue developing. HCG also stimulates the developing testes to produce testosterone in male fetuses. This will ultimately result in the lowering of the testes into the scrotum and the development of male sex characteristics. Once the placenta has had about 7 weeks of development, it can now produce progesterone. This means that the corpus luteum no longer has a role to serve in embryonic development, so it disintegrates. The placenta now produces both HCG and progesterone. Peak HCG levels are produced around 3 months, and then they decline. HCG is typically the hormone that is detected during pregnancy tests. 2 additional hormones that are present during early embryonic development and throughout the rest of pregnancy are estrogen and progesterone. Now, we've already mentioned the importance of progesterone in 2 key roles. First, we discussed how progesterone relaxes the smooth muscles of the fallopian tube as the zygote travels from the tube to the uterus and stimulates blood vessel growth in the endometrium upon egg fertilization. Then we discussed the critical role progesterone plays in keeping the endometrium of the uterine wall intact to prevent contraction and the shedding of the endometrium and in turn the developing embryo. Progesterone, along with estrogen, is necessary to prevent the development of another set of follicles. These 2 hormones suppress feedback to the pituitary, which prevents this development. Estrogen is also necessary for the development of the milk-secreting ducts in the breasts. The final hormone we are going to discuss is human chorionic somatomammotropin, or HCS. Like HCG, estrogen, and progesterone, HCS is secreted by the placenta. It is closely related to prolactin, which is a hormone that's important for lactation. It's likely that HCS plays a role in lactation, but its presence is not necessary for lactation. HCS is a major contributor to maternal glucose and fatty acid metabolism. Glucose enters fetal circulation after it's transported across placenta membranes via facilitated diffusion. Unfortunately, a small percentage of women develop gestational diabetes during pregnancy. Gestational diabetes is similar to type 2 diabetes in that it is marked by insulin resistance. Typically, blood glucose levels return to normal postpartum, but gestational diabetes does have implications for both the mother and the baby later in life. As you can see, hormones are critical in the implantation of a blastocyst and in the development of an embryo. Without progesterone, the zygote would have trouble traveling through the fallopian tubes to the uterus. There would be insufficient endometrial blood vessel development, and there would be insufficient nutrient secretion to the embryo from various glands. Additionally, a drop in progesterone could result in the contraction and the shedding of the endometrium. This results in the loss of the embryo. HCG is necessary to maintain the presence of the corpus luteum, and it's necessary for the proper development of the testes and sex characteristics in male fetuses. Estrogen plays an important role in the prevention of new follicle development during gestation, along with progesterone, and in the development of milk-secreting glands in the breasts. Lastly, HCS is important in the metabolism of glucose and also in fatty acids.